Hi everyone, this is 12th video in our Advanced Pharmaceutical Dissolution Science and Application series. In this video, we will discuss dissolution in QC versus R&D versus stability, what changes and why. Dissolution testing is not a single purpose test. It evolves across product life cycle, for exploratory R&D to routine QC and finally long-term stability. Each phase has different objectives, precision requirements, robustness expectation, and regulatory constraints. In this session, we will understand how dissolution testing changes across R&D, QC, and stability, and why each stage demands a different approach. Dissolution testing is multifunctional. It serves three distinct purposes across the product life cycle. In R&D, it supports formulation development and understanding. In QC, it ensures batch-to-batch quality, and in stability, it monitors product performance over the shelf life. Because these goals are different, the acceptable variability, method flexibility, and sensitivity also differ at each stage. So even though the test looks the same, the purpose changes, and that drives different expectations. In R&D, dissolution methods are exploratory, not official. Teams freely adjust pH, medium apparatus, agitation, and surfactants to understand how the formulation responds. The goal is to screen prototypes, study API solubility and release mechanism, and optimize the formulation. That's why high sensitivity and strong discrimination are critical in R&D to detect even small differences between formulations. In short, R&D dissolution is flexible, investigative, and learning focused. In R&D, dissolution method can be adapted rapidly as new data emerges. For modified release products, teams often use USP apparatus 4 to understand release mechanism under controlled flow conditions. In early development, mini vessels are commonly used to reduce API consumption. R&D methods are experimental and may use biorelevant or non-standard media with a wide pH range explored. Typical R&D practices include use surfactants and biorelevant media, multiple apparatus USP4, USP2 and mini vessels, dense sampling time points for deeper mechanistic insight. This flexibility is what makes R&D dissolution a powerful learning tool. In quality control, resolution methods are logged after validation. There is no flexibility. The focus shifts from learning to consistency. The method must work every day for every batch and for every analyst. In QC, priorities are routine batch release testing, a validated and fixed method, robustness, reproducibility, and precision. Minimal variability between vessels, runs, and analysts. Here, consistency matters more than sensitivity. In quality control, resolution testing relies heavily on automation, daily calibration, and repeatability. QC uses a single, validated, and fixed method for product with strict SOP adherence and minimal variability. Key QC characteristics include tightly controlled media and apparatus, limited sampling points typical 1 to 3, stricter system suitability criteria such as percentage coefficient of variation less than 6% at stage S1, emphasis on robustness and precision, not exploration. The goal is simple, consistent, reproducible results. Every run, every day. In stability studies, resolution shifts from a release test to a monitoring tool. The same validated QC method is used, but the expectations change. Here, we closely trend the dissolution profile over time to detect performance drift across the shelf life. Stability dissolution helps identify change in tablet hardness, polymorphic transformations, moisture uptake, excipients degradation, and gradual shifts in drug release behavior. Compared to routine QC, Stability testing applies higher scrutiny and sensitivity because even a small trend can signal long-term risk. In short, stability dissolution protects product performance throughout its life cycle. Across the product lifecycle, the resolution method must balance discriminatory power and robustness. Each stage has different tolerance for variability. R&D prioritizes high discrimination to detect formulation differences. QC demands high repeatability and minimal variability. Stability requires high sensitivity to trend small changes over the time. So, while the test may look the same, the purpose is different at each stage, and that purpose drives method design. What you prioritize in R&D, QC, or stability must change accordingly. Media flexibility changes across the product life cycle. In R&D, flexibility is highest. Teams may use non-physiological pH, surfactants, and biorelevant media like FASSIF or FESSIF to understand formulation behavior. In QC, flexibility is very limited. Only validated and approved media can be used. FASSIF or FESSIF are not acceptable unless they are fully validated and regulatorily justified. In stability, the focus is consistency. The same media as QC must be used because even small pH or media changes can mask or exaggerate true performance drift. So remember, R&D maximum flexibility, QC strict validated media, stability identical media for reliable trending. The purpose of testing defines how flexible your media choice can be. Apparatus selection also changes across life cycle. In R&D, teams may explore multiple USP apparatus, including USP 1, 2, 4, or 7, depending on the product and the question being answered. This flexibility helps understand the release mechanism early in development. In QC, focus shifts to simplicity and repeatability. Most products use USP apparatus 2 pedal or sometimes USP1 basket. In stability, the rule is simple. Use the same apparatus as QC. Consistency is essential for reliable trending over the time. So remember, R&D flexible, exploratory apparatus selection, QC simple, approved and repeatable, stability identical to QC. The stage of development defines the level of flexibility. Sampling strategy also changes across the life cycle. In R&D, dissolution profiles are information rich with 8 to 12 sampling points commonly used to understand release kinetics and mechanism. In QC, dense sampling is not practical or necessary for routine batch release. QC typically uses 1 to 3 validated time points, focusing on efficiency and consistency. In stability, the same time point as QC, but the focus is on trending results over the time, not adding more samples. So remember, R&D rich profiles for learning, QC minimal points for efficiency, stability same points tracked over shelf life. The purpose of testing defines the sampling depth. From a regulatory perspective, dissolution method must balance robustness, discriminatory ability, and change control. In R&D, methods are flexible and exploratory. Formal validation is not required. However, R&D data must eventually justify the final QC method. In QC, a fully validated method is mandatory, in line with ICH Q2R2. This validated method is then logged and approved. In stability, same validated method as QC is used to ensure consistent long-term trending. Any method change after approval requires scientific justification and may trigger a regulatory filing. So remember, R&D flexible, no formal validation, QC validation mandatory, stability uses the validated QC method. Post approval changes controlled and justified. That's how dissolution method move from development to regulatory acceptance. Understanding how dissolution evolves across the product life cycle helps analytical chemists design better, more robust methods. Remember, the core idea R&D is exploratory and flexible. It helps to learn. QC is fixed and robust. It helps to control. Stability is monitoring and sensitive. It helps to detect change over time. It's the same resolution philosophy, but the different applications at each stage. When you design method with this life cycle perspective, you build resolution tests that are scientific, practical, and regulatory ready from development to end of shelf life. R&D learns, QC controls, stability monitors. Three phases, three goals, one test. Thank you for watching. If you found this explanation helpful, please like, share and subscribe. From this video onward, we are just finishing our core fundamental understanding on dissolution, and in next section of 10 videos, we will just understand dissolution method development for OSD product. In the next video, we will cover step-by-step dissolution method development for immediate release tablets. And stay tuned because in this 62 video series, we will dive through resolution method development, validation, troubleshooting, and advanced approaches and real industry case studies. If you have any question, please post them in comment section. See you in next video. Thanks.